In this screencast, we're going to be looking at DNA structure, and we already know the DNA structure was discovered by, or at least that's where all the credit goes to, Watson and Crick. And they used modeling. They actually used 3D models and a bunch of other information that we're going to talk about here in this screencast to come up with the three-dimensional structure of DNA. But they had a lot of help and a lot of other information along the way that got them to this point where they could actually elucidate the structure. So let's take a look at some of the history of discovering DNA. All right. So there was a scientist called Frederick Griffith, and in 1928, he did an experiment where he had pathogenic bacteria that he knew that if he injected into mice, it would kill the mouse. He also had another strain of bacteria that was very similar, but it was non-pathogenic, that if he were to inject that into a mouse, it would be fine. Well, he took some of the pathogenic S bacteria and he killed it with heat and injected it into a mouse, and sure enough, the mouse lived because the bacteria was dead. But then he took a combination of the heat killed, which should give a healthy mouse, which shouldn't kill it, and the non-pathogenic bacteria, which shouldn't kill it, and he mixed those two together and gave it to the mouse, and the mouse died. So he figured there was some sort of chemical component, some genetic component that the non-pathogenic bacteria were getting from the heat-killed bacteria. And sure enough, when he sampled them, he found something that was more like these living pathogenic S cells. He called this genetic passing from these living bacteria from the non-living ones, he called that transformation. And we're actually going to look at transformation when we do biotechnology later on. So he coined that term. All right, the next study we're going to look at is a study that was put together by Hershey and Chase. And they were using viruses and bacteria to figure out what was the genetic material, because it wasn't known at the time. Was the genetic material proteins or was it DNA? Those were the two suspected organic molecules. Well, let's talk about what we know about viruses. So this is a T4 phage that is a virus that will infect bacteria. And the virus is basically a protein coat with DNA, and it'll inject its DNA into the bacteria. And then the bacteria will use that DNA information to build more viruses and end up killing the bacterial cell. And so essentially all the virus is made up of is a protein coat and DNA. Well, of course, they didn't know exactly which components were the genetic material at this point. They just knew that the bacteria was going to be infected with genetic material. So they designed a test to be able to figure out, is it the protein of the virus that's the genetic material, or is it the DNA? So let's take a look at their study. It's pretty complicated, but we're going to look at it step by step, and you're going to have to answer some questions about it in your textbook. All right, so here is the Hershey-Chase experiment. They had a batch of bacteria and viruses that the viruses, what they did with these viruses is they, this first set, they inoculated with a radioactive sulfur, okay? And we know that there's lots of sulfur in proteins. They make those disulfide bridges, especially when we're looking at the tertiary structure. But there's not sulfur in DNA. They already knew that, what the chemical components of proteins and DNA were. They just didn't know which one was the genetic material. So they had a bunch of viruses that they cultured in a radioactive sulfur. So they knew that all of the protein had this radioactive sulfur, so they could track the protein. And then a second batch of viruses, they inoculated with a radioactive phosphorus. And we know that there's phosphorus in DNA, but not in proteins. So they knew that they would be able to track the DNA in this second batch. Again, they didn't know which was the genetic material. Was it proteins or was it DNA? So then they took these radioactive viruses and they exposed them to bacteria. And then they knew that the viruses were going to inject the genetic material into the bacteria, which happened. And then when they were done, they they ended up putting into a centrifuge the remaining bacteria in the solution that they were being exposed to the viruses in. And in the centrifuge, after it had been centrifuged, they knew that the casing, what was left over from just the virus, would be in what we call the supernatant, and that anything that was injected into the bacteria would be down here in the pellet. So we knew anything that got injected, anything that was inside the bacteria would be in the pellet, and anything that was just part of the casing and not into the bacteria would be in the supernatant. And so when they did that to both of these, they realized that all of the protein, or most of the protein, was in the supernatant and not in the bacteria. They noticed it was on the outside. And when they ran it with the bacteria that had been infected with the viruses with the phosphorus, then when they ran it through the centrifuge, they saw that all of the DNA was actually inside the bacteria and not in the supernatant. And so that allowed them to realize, oh, well, it is actually DNA that we know that the viruses are injecting into the bacteria. So they said, oh, the genetic material must be DNA because of the T2 phage. All right. And this is a set of questions we're going to have you guys answer on this experiment. So you're going to have to be able to talk about the supernatant, that's the liquid part, and why the genetic material should be found in the pellet with the bacteria rather than in the supernatant. And then you're going to read some graphs to figure out how much is left in the supernatant and how much of the DNA is found inside the cells and so on. Okay. So that's on page 344 and 345. If you want to pause it, you can go work on that now and then come back to the rest, or you can just keep watching. Okay. Another scientist that was important in the Watson and Crick discovery was Chargoff. And this happened actually before the Hershey Chase experiment. And Chargoff realized that DNA composition would vary from species to species. And so he broke down kind of the amount of different nucleotide bases you could find in different species. So he would take a species and he would count like what proportion of thymines existed and what proportion were adenines, what proportion was cytosine, and what was guanine. Well, what he found out, which of course makes sense to us now since we understand so much about DNA, is that within a species, if let's say it was 10% thymine, it would always be 10% adenine. And then the other 80% would be 40% cytosine and 40% guanine, making up 100% of all the nucleotides, right? If it was 20% thymine, it would always be 20% adenine. And then, of course, the 60% left would be 30 of cytosine and 30 of guanine. So there was always the same proportion of A's and T's together, and there was the same proportion of C's and G's. So that gave Watson and Crick an idea that we know that there needs to be the same number of thymines as adenines when putting together the model. Something else that you're going to start to learn about too that we know about DNA now is you can see some slight differences in the nitrogen base, the sizes of them. We have things called, the adenines and guanines are called purines, and the thymines and cytosines are called pyrimidines, and you notice there's a little bit different in the size of those nitrogenous bases, and that affects the width of the DNA. Okay, so Chargoff discovered that the ratio of thymine to adenine, that was the same ratio, and the cytosine to guanine was the same ratio, making up 100% of the nucleotide sequences. And then the next part was when we knew that DNA was probably the genetic material, Rosalind Franklin and Maurice Wilkins started working on a technique called X-ray crystallography, and they wanted to study molecular structure. And there were some other scientists working on this stuff too, like Linus Pauling. But Rosalind Franklin was working at King's College. She got the absolute best picture of the DNA molecule ever known. It was beautiful. So she got this perfect X-ray crystallography picture of DNA. I'll show you what that looks like here. So here's her image that she managed to get of this. It's her diffraction photograph. And people were pretty excited about this, but she didn't want to publish until she had enough evidence. But some of the things she was able to figure out from this was that it was a helix, right? It was helical shape. And that the steepness of the angle, so she knew how steep the pitch was when it was spiraling. And also the distance between the horizontal bars, she knew what the width is, the diameter, and it was a consistent width all the way down. So she was able to come up with a whole bunch of mathematical pieces that gave us an idea about what the structure was. Well, between Rosalind Franklin's ideas and Chargoff's and so on, that's when Watson and Crick started to build their model. And it turns out actually that this picture, which Rosalind Franklin had not published, was actually given to Watson without her permission. And he was able to take that information to help him build this. And it's actually, she's really, really well known. And a lot of people think, well, of course, she should have had the Nobel Prize like Watson and Crick. But unfortunately, by the time her role was accepted and more prominent, she had already died actually quite young. And it's likely a lot of the things that she was using in her research were probably what killed her in the end. But she died quite young at I think 37 or something like that. She died very young, and you can't get the Nobel Prize if you are already dead. So by the time that would have come about, she was no longer alive to receive it. But Watson and Crick used her information about the steepness of the angle and the width of the molecule to be able to figure out, which one of these models go together. So they knew that it was helical in structure, and they could deduce the width of the helix because of the spacing of those nitrogenous bases, and that it was actually made up of 2 strands. So this is kind of what we know the structure to be now. And one of the interesting things that they were able to figure out, especially with the width here, is that, remember I had mentioned to you about the purine and pyrimidine. Well, they then knew that 2 pyrimidines together would have been too narrow for that picture, and 2 purines would have been too wide. So they knew that it had to be a purine paired with a pyrimidine. And then, of course, with Chargoff's information about A's and T's having the same, proportion, that's how they sort of knew that, oh, well, it has to be A to T and C to G. And then that worked out with the bonding. And you can see there's 2 hydrogen bonds between the adenine and thymine and 3 between the cytosine and guanine in it. And it creates that nice, stable double helix. All right. So I am going to end our little discussion on DNA, the discovery of DNA structure.